So let's take a minute here and look at a case study. In this case, the disease known as Duchenne's muscular dystrophy. And we're going to look at this disease because it's a really good way to illustrate some of the points that we ask you to think about in terms of mutations, types of mutations, and the consequences. So briefly, what is Duchenne muscular dystrophy? Many of you have probably heard of it, but essentially it is a disease that manifests in muscle weakness and wasting. What happens is that normal muscle structure does not develop, and as a result, muscles atrophy, muscle connections aren't made effectively, and a variety of other issues can arise. And you'll notice that males tend to demonstrate this type of disease more than women. So think back to the last unit, and if you have a situation where you have a human disease that appears more in men than in women, what does that tell you about the way that it's inherited? I'm going to let you think about that on your own. So let's move specifically into muscular dystrophy. There's a gene called dystrophin, and this is really what is going on in the background of patients with muscular dystrophy. There's a gene called dystrophin, and there's a protein called dystrophin. And from what you remember about how the Central Dogma works, it is a sequence of DNA, a small sequence of DNA, the gene, that is then transcribed into mRNA. The mRNA is then translated into a protein, and in this case, the protein is called dystrophin. This gene is found on the X chromosome. So again, think back to last unit. If this is found specifically on the X chromosome, why do men seem to demonstrate this disease more? So anyway, multiple mutations can lead to the phenotype of muscular dystrophy. So we're going to look at the gene itself. The dystrophin gene is very large. It's about 3,500 amino acids that come out of this gene. And there are 79 exons. Remember the difference between exon and intron. You can review that on your own as well. So we're focusing specifically on the dystrophin gene, which is a very large gene that encodes for a very large protein. This large protein provides a bridge. You can see here dystrophin in red provides a bridge between different muscle fibers and how they contract and how they join together. So it is an integral part of proper muscular function. So if we look here, we have a strand of DNA. You can see it's double-stranded because you have a 5' and a 3' end in a series of letters. So what we have here are a series of letters in black, which mean we're not going to pay attention to them, and then those that are in blue. So consider this. And for this case study, I'm not going to tell you the answers right now. You're going to need to think about what's going on here. So if we consider the above DNA sequence, this blue area here has a mutation in it. So what happens is this mutation will ultimately, during some process of the Central Dogma, lead to a stop codon. So what this question is asking is, when a DNA polymerase, which is this blue box here, reaches the nucleotides encoding for a premature stop codon, what will happen? I want you to write down your response and think about it. So again, we have double-stranded DNA. We have DNA polymerase working through here. What's going to happen when it reaches this mutated area? Okay? Now we're going to move to another scenario. Again, when I progress, make sure you take notes on the next scenario and write down your predicted response. We have the same double-stranded DNA sequence. We have the same mutated section. In this case, we have this red box here, which represents an RNA polymerase and the process of transcription. Again, ultimately, what this mutation is going to lead to is a stop codon. So, as the RNA polymerase progresses along this template strand, what is RNA polymerase going to do? So this question, same double-stranded DNA, RNA polymerase moves along this template, what will happen? Write down your response and reflect on it for a minute, and then there's one more question coming up. Okay. Finally, we have, again, the same sequence here of double-stranded DNA. The mutation ultimately leads to a stop codon, as we've said repeatedly. In this final question, I'm asking you to think what will happen when a ribosome reaches the mutated bit. So write down what you think is going to happen this time. Now we have a ribosome. What is going on here? Okay, so you should have thought about these three questions and written down your particular responses. So what's the effect of a single mutation leading to a stop codon on a muscular dystrophy patient? So as I've told you before, the phenotype is the disease muscular dystrophy, which is an atrophy of muscle fibers, the incorrect formation of muscle fibers, or incorrect movement of muscles in a typical fashion. So think back to the three questions I asked, and also think back to this small module on mutations. What type of mutation ultimately leads to muscular dystrophy? For those of you who stayed around, I'm going to answer the questions for you now. So the first question asked, what would happen when DNA polymerase moves along here? Remember several things about a polymerase. It reads one letter at a time. If this ultimately becomes a stop codon, this point mutation, does DNA polymerase actually matter here? DNA polymerase is just going to go through one letter at a time and make a complementary strand of DNA. This is DNA replication. Therefore, a stop codon has nothing to do with anything. All DNA polymerase is doing is replicating DNA, so nothing will happen. The second question, this is dealing specifically with transcription. So we still have this mutated section of DNA. RNA polymerase is transcribing, again, one base at a time. As a result, when it reaches this mutated bit, it just creates a complementary mRNA strand. So nothing happens except RNA polymerase makes mRNA. Finally, what will happen when a ribosome reaches a stop codon? This is where the effect happens. It happens in the synthesis of the protein, and it happens in translation. Ribosomes read codons. Codons cannot be read by polymerases except to just make a complementary base pair because they read one letter at a time. Codons are these three letters here, and a ribosome reads a codon. So when a ribosome reads a codon and it codes for a stop codon, it falls off. So what's the ultimate effect? You have a protein that is too short. If you have a protein that is too short, it's because a stop codon occurred during a mutation. So what type of mutation leads to muscular dystrophy? it would be a nonsense mutation because a stop codon was created by a single base pair point mutation.